Is this the exact question, or are there any differences in what you had and what is here?
Same, sir.
Okay, good then. So, which parts did you understand and which parts didn't you understand?
There was no information about the probe in the given question, sir.
Okay,
Yes,
here I have added then. Okay, probe is made based on sequence of gene P. So you should assume that it will bind somewhere here like this. Okay, 1 second. You need a little more information is there. So what you're supposed to do is also there, right?
yes, sir.
Yes. So this is what you should do. Oh, okay. But otherwise, did you understand the rest of the sentences? So there is gene P. Let me try to go through and let's see what we understand. Gene P and gene Q are two adjacent genes and imprinted. So, and one is methylated paternally and one is methylated maternally, right? Assume that all imprints. Mm, 1 second. Okay, we will see if there is any question in here. Where is it? Assume that all imprints are erased during primordial germ cells and established during gametogenesis. There are restriction sites, and these are the dotted lines that are given. And then there are those sequences could be recognized, those sites could be recognized by three different restriction endonucleases, X, Y, Z. X and Y, they have difference. X is methylation dependent, and Y is methylation independent, or sorry, insensitive, and Z is methylation sensitive. Okay, now you have to solve. What is there that you didn't understand? Tell me. Based on the experimental setup, biological assumptions that you know, and the data that is given here, 11 kb fragment is already given to you. Predict the banding pattern in each of the lanes given as above, I mean here, and imprinting patterns of gene P and Q. So first thing, gene P is paternally methylated. Okay, we don't know which one is paternally imprinted, which one is maternally imprinted. Gene P and Q are given, and one of it is maternally imprinted and one of it is paternally imprinted. Okay, can you try to solve it? And ask me if you have any questions, and then this is trying to go along with you and trying to solve it. Otherwise, it'll be one way and just saying the answer if there is one. But the process of how we get from the question to the answer, the process is more important than the answer itself. So you can ask me any questions that you have.
Sir, it is given in sperm using Z restriction enzyme. We are getting 11 kb fragment.
Yes.
But what happens to the 4 kb fragment, sir? It is also.
You tell me.
Sir, I couldn't understand. The Z
It,
is actually methylation
it doesn't
sensitive.
matter, right? Here, this is the probe is limited till here. You will perform southern blotting, right?
Yes, sir.
Probe, whatever probe is binding, that is the only fragment that will be detected, isn't it?
Yes, sir. Yes, sir.
Okay. Now tell me.
Sir, we have to consider like gene P. It is our choice to choose that is paternally methylated or maternally.
No, you cannot choose like that. If how I think that what you should think is how can you get this? If what kind of situations will give rise to this kind of band here? In this sample, With this restriction endonuclease, you got one band, right? So what will determine that?
So it
What?
means gene P is paternally imprinted. Uh, Gene Q is paternally imprinted, sir.
Mm, I'm looking at if there is a mistake. Let me check. Mm, I will
Sir,
change it to something. Okay.
sir, it is gene P only, sir.
No, no, you have to think a little more. Okay. For ease, I will do one thing here. Assume that it is 15 kb fragment. Okay. You understood?
Yes, sir.
Assume it is 15 kb fragment. So now you try to decide what it is.
Sir, first we have to clarify about the restriction enzyme, sir. What is methylation dependent? And,
Oh, did we never discuss that?
we discussed, sir,
Okay,
but
then it's all right. That's all right. Okay. So if there is a restriction site, say there is a restriction site here, and that region, okay, I will just take a real example. For example, it's like GAA TTC, GAA TTC. This is a restriction site. So if an enzyme like this is for EcoR1 can cleave it like this, then we say if it can cleave, it is a restriction endonuclease. But as per the way we are learning the epigenetics, we know about DNA methylation. So DNA could be methylated. These sites could be methylated like this. If a restriction endonuclease cannot cleave, if this restriction site is methylated, then you call it as methylation sensitive. That means it will not it will not cleave if the restriction site is methylated. If there is a restriction endonuclease, it will cleave irrespective of whether this restriction site is methylated or not. Then you call it as insensitive. These are just our terms. Okay, did you understand this? Methylation insensitive. Say why restriction endonuclease Y will cleave this site irrespective of whether this site is methylated or not. That is methylation insensitive. Whereas Z, for example, it will cleave only if this methylation, if this restriction site is unmethylated. And there is X will cleave this site only if it is methylated. If this region is not methylated, then it will not cleave. Is that understood?
yes,
Yes.
sir.
Okay, you have any question on that, you can ask. This is a theoretical problem. Okay, it's about how we are thinking and trying to apply biological concepts. I mean, use these logics for biological concepts. Yes. Do you have a question? Okay. Did you figure this out? In what situations will you have 15 kb fragment? How will you get 15 kb fragment? In which conditions will you get 15 kb fragment? We
If
will get,
gene
yes, go ahead.
if gene P is not methylated, we'll get
Yeah,
around.
if this part is cleaved here, okay, if this is cleaved, then this is a 15 kb fragment, right? That means if it is cleaved by what is Z, Z is methylation sensitive. It will cleave only if that region is unmethylated. So what can you, this is sample is from sperm. So gene Q is it maternally methylated or paternally methylated? Yes.
Paternally related.
Why do you say that? This region, okay, is it maternally or paternally?
Sir, I have another doubt, sir.
Okay.
Sir, gene P and gene Q, it doesn't have to do anything with sperm or egg.
I said they are methylated. One of it is maternally imprinted, one of it is paternally imprinted.
No, sir. These two are present. These two genes are present in either case, that is sperm or egg. In both cases, it is present.
Yes, yes.
Then, okay, sir.
All you need to remember is in egg, it is N. Somatoplasm, diploid. Primordial germ cells, diploid. In sperm, it is haploid. Okay. Okay, let's. What can you tell about gene Q?
Gene Q
In
is
this
maternally
region?
imprinted, sir.
Yes, it will be maternally imprinted. How do we say that? Only then we have gene Q, sorry, P, and these are the restriction sites. We are getting a 15 kb fragment. That means this region is not methylated because we are talking about Z, which will cleave only if that region is unmethylated. So gene Q is maternally imprinted. That much we can clearly say. And from there, you can work out the rest of it. Can you do that? What will be with Y? What band would you get in Y in this same in sperm?
We'll get 4, 11, and 7.
Yeah, but only one band will be visible. You should tell me which band is it.
11 kb.
That's right. It will be 11 kb fragment. With
Sir, with
X,
the,
yes,
now I figured out, sir, which was confusing. Probe
yes.
information about the probe is not given, sir. That is.
Okay, that we have given now. What about X? With X, what would you get here?
15, sir.
Why is that?
Sir, because the cut will be only between gene P and gene Q.
Yes.
So we'll have
What is X? X is methylation dependent. So it would have cleaved. If this one we are assuming in sperm, we are assuming this is not methylated, we would have assumed this is methylated, right? And X will cleave only where there is methylation. Yes? If you have a doubt, you can ask. Then I'll try to see if I can explain it differently.
sir, can you repeat the same thing?
Okay. So let's check this out. We have Q, we have P, and these are the restriction sites. How did we get, if we had this 11 kb fragment, what does that mean? In sperm, and with using Z, we got 11 kb, 15 kb fragment. That is possible if only there was a restriction cut here, isn't it? This is where the cut is made. So then that means this region is unmethylated. Yes, you agree with that? This is not methylated. That's why Z was able to cleave there. If you have, which would mean in the case of Y, methylation insensitive, it would have cleaved in all the places, right? What I'll do is, for ease, I will try to do it differently. Just stay with me. I'll try to give three different conditions, Q and P, and we have these fragments 4, 11, and 7. So what will happen if we cleaved with 7, with Z, we got a fragment that is 15 kb, and this one will be 7, which will not be visible because it will only cleave, Z will cleave only where there is no methylation. With Y, it will cleave irrespective of whatever it is. It doesn't matter whether it is methylated or not, it will cleave. So we should get 4, 11, and 7. Only this will be detected. And then we have another one, that is this. With X, X is methylation dependent. If you're assuming this is methylated and the question says that one of it is maternally imprinted and one of the other one is paternally imprinted, we should assume that this is maternally imprinted. So we'll assume then this is paternally imprinted. That means this region would have been methylated, which means X would have cleaved this DNA only here, which will result in the fragments 4, 11 plus 7, 18 kb fragment. Did you understand that? And this will be detected.
Yes, sir.
Do you have any questions on this? Yes?
Understood, sir.
Okay. Then what do you think we should do?
18 kb fragment.
Yes, so we will get 18 kb fragment. Now we can try with the egg. That is easier. So what would you expect with Z? With Z? In egg, again, this methylation is fixed, right? One is maternal and one is paternal. Now the sample is changing. The sample is coming from egg. So what would you expect in that case for gene P and Q? You should tell. In sperm, one of it will be methylated, one of it will be unmethylated state. Whereas in now it is in the egg, so we should reverse those things. In the egg, let's see. In the egg, maternally imprinted, we are saying this one. So we should have had methylation here, and this should remain unmethylated, PQ. And if you then treated this DNA with Z, we would have cleavage. We would have had cleavage here, right? Do you agree? Because that is the only unmethylated place. So we'll have cleavage there. And then we should be getting a fragment that is of 18 kb. If we treat it with Y, which is methylation insensitive, means it will cleave irrespective of whether methylation is there or not. So we will get a fragment like this, 11 kb. And if we treated it with X, then we would have
15.
Yes, we will get 15 kb fragment. Right? Then now you can solve for somatoplasm and primordial germ cells. What else you have to consider is in somatoplasm we have 2N, and also in primordial germ cells they are biparental. So we actually have to consider more things, right? If it is biparental, we will have something like this. So gene Q will have P. One of it is inherited from the paternal allele, the other one from the maternal allele. So what we have determined from the given data, this one is that Q is imprinted from the maternal allele, P is imprinted from the paternal allele. I hope that is correct. Yes? I'm showing these as darkened ones as the um, methylation. So now if we treat it with Z, which is methylation sensitive, that means it will cleave. What are the fragments? Can you tell me? 4,
Sir, for
11, 7. Yes, go ahead.
PGC you're asking, sir.
For somatoplasm. For somatoplasm here. If we cleave with Z, it will cleave only where there is no methylation. So the fragments would be here 7 and 15, right? It will cleave 7 and 15 fragments, but only 15 will be visible because that is where our probe is. This is for one set. What about the bottom one? It will cleave for the maternal allele. It will cleave in this location, but not here because that region is methylated. So what will you get then? 4 and 18. So what you will be able to visualize is a 15 kb fragment and also 18 kb fragment. Okay.
Yes, sir.
Ritika, can you help me with Y and X?
I will try, sir.
Yes, tell me. Y is
11
methylation.
kb for both, sir.
You get 18 and 15, sir.
Okay, what is Y? Methylation insensitive. It will cleave irrespective of whether the sites are methylated or unmethylated. Right? So
I
they will
guess
cleave.
for all we'll get 11 kb fragment only, sir.
That's right. I think you will only get one level kb fragment because both you will get 11 from here as well as here. Both are same size, so it will form only one band. Okay. What is from X? X is
18 and
methylation
15.
dependent. So what will you get? Go ahead.
For male we will get 18, sir.
Yes, 8, 18, and another
15, 15, sir.
15 from the maternal allele. And then in primordial germ cells, what will be the methylation status in primordial germ cells?
For Y we will get 11 kb, sir.
Primordial germ cells, the erased imprints are erased. Okay, tell me now. For what will you get for Z, Y, and X?
11, sir. For Z we will get 11.
For Z will get 11. Okay. Then
Y
for
also 11, sir.
Y will get 11. Okay.
For X we will get whole, sir. Whole 4 plus 11.
Yes.
4 plus 11 plus 7, sir.
Say it again.
Sir, we will get holes.
Whole, what is the total?
22.
Yes, we will get 22. Tell. Yes, sorry. What was I telling?
22.
Yes, this is 22. Now, did you understand the question?
Yes, sir.
Okay. Yes, it was a mistake on my part that this is one thing that I should have made it correct. Ritika, are you clear or you have any questions on this?
So it's a little bit confusing, sir. We'll see
Yeah,
once.
there are too many things in this. We need to think about many things, isn't it? That's why it's very common to be confused. We need to know about restriction endonucleases, methylation dependent, and all. We need to know about where the probe is located. We need to know the sizes, and we need to know the sample, and we need to keep track of imprinting. In sample, we need to know if it is haploid or diploid. And in imprinting, we need to know if it is maternally imprinted or paternally imprinted. And if it is diploid, we should know if it is somatoplasm or primordial germ cells. In one case, you have imprint established and biparental. In another case, imprint erased. So we need to take into account there are too many factors to keep track of. And that's why this is one of the favorite questions I do have. It'll be, I mean, this is complex, it's puzzle, right? I only give one thing and the rest of the things you have to do. You sometimes I give the data and I ask you to determine the imprinting status. I will not give many other data. Okay, so I might ask you to determine the which one is maternally imprinted, which one is paternally imprinted, and so on. So it's all right. What you need to be clear is when you're changing from one lane to another lane. Say, I'll try to erase a little bit here. So with this sample, you need to remember what you should be doing, right? If it is X, Y, sorry, Z, X, Y, Z, you need to know how they are different. And you should always use a lot of paper, rough paper, trying to draw these things on paper. The more you think on the paper, you will get better. And that is, I think, I mean, that will be have some clarity for solving problems like this. Okay.
Yes,
But
sir.
if you have any other questions, you can ask me though.
Sir, next question, sir.
Why would I tell that?
Sir, we will tell, sir. Maybe you can verify. We
No,
can
you are
verify.
supposed to answer. You will upload it, isn't it?
Yes, sir.
No.
Yes, sir. Yes,
Then
sir.
when you upload, I will know. Okay, what is your answer so far? You tell me that, and then I know I will know. What is your answer so far on that?
Sir, for this given diagram, the RRS is opposite, that is inversely directed. So we have to. So this will give inverted products. Sir. That is the between RRS, the genes will be inversed. So
Yes,
we'll get
inversion will occur.
so we'll
Yes.
get gene Y to the left, and
Okay.
entity will be.
So there are two things. One thing is the complete question of it would be something like comment on the DNA. First thing is that comment on the construct, and then you are supposed to design a good one, the correct one. What do you think is the correct one? Tell me, what should I do? Tell me the gene names, I will write.
Sir, that is you want gene X, gene Z, gene Y alone and not others, sir.
Yes. So the original one, the in the there it is X and Y, and what we want eventually is we want to have X, Z included, that is the human gene. Assume that we want to insert gene Z between X and Y and nowhere else. Okay, that's the full question. We want only gene Z to be integrated and nothing else and nowhere else. So how do we get that?
Sir, is it like we can change the RRS?
You can do anything you want. You can reorganize the whole thing also. I need to get this is the output I want. How will you do that?
Sir, I have one, sir. First, we invert this as it is, and we'll get
You
gene
want to
Y.
put it this way, or what is it? What do you want to do?
No, sir. As a result of this indirect repeat, we'll get NPT and gene Y to be inverted, sir.
You want this NPT to be inverted. Tell me again. I'll try to write now.
Gene X.
So what do you want to do? Okay, maybe I'll write it here. What should I do to NPT? Should I put it this way? That's what you meant? I didn't understand. You can tell me, please.
No, sir. The NPT and gene Y, the place will be, it will be reversed, sir. Gene
This
Y
way?
will come to the left and NPT will go to the right.
Okay. Do you want this one to go here? That's why what I was asking is you tell me the genes, what should be there from this end, and then I'll keep
Yes.
writing, and then we will know. What
Gene
should I do?
X prime,
Okay, X, then,
then gene Z,
okay,
then
Z,
gene Y prime, and then NPT.
then gene Y, and then NPT.
NPT, and then TK.
Okay, what about the arrowheads? What are they? Okay, what is yours, Marzuk? You tell me, please.
Sir, mine is also same, sir. But after inversion, I'll go for deletion, sir. That is forming RRS between NPT and TK. That is
Hmm.
after that we'll get gene Z Y X.
Okay, if you have, you can post whatever you wrote. Then I can see and know where you are standing, and then I can probably answer it properly. Can you WhatsApp it to me?
Sir, I didn't write it up.
Ah, then you haven't solved it.
Sir, I solved in rough, sir, but I
Where is
didn't.
it? That is fine. Rough is fine. I'm good with rough. Send it as it is. Sending?
Yes, sir, will send.
Okay.
Sent, sir.
Yes. Where is your answer? X, Y. Okay. Yes. Oh, you wrote the same answer as well. You got the
I'm
same
not
answer.
complete yet.
Huh? Yes.
I'm not complete yet, sir. I have to eliminate NPT and
Okay.
TK.
What we will see, what we will discuss is why do we have in the DNA construct, why do we have gene X? First, if you can tell me why do we have gene X? Gene G is the purpose. We want to put that particular gene in here. Okay. Why do we have NPT? Why do we have gene Y? Why do we have TK? What are the purposes of it, of these genetic elements?
NPT is for a positive selection. The cell may the gene is prioritized, sir. So we
This is positive selection. Okay.
TK is negative selection.
Okay. Negative selection. Okay.
And
Why do we have?
X
Uh,
prime and Y prime are homologous regions of the.
yes, this for homologous recombination, we'll say. Okay. What are these arrowheads? Why do we have them?
So the inverted repeats.
Okay. What are they useful for?
So homologous recombination.
No. This is for site-specific recombination. What I will do is I think you have to I'm assuming I'll just switch off the recording first. Just give it a minute.